Hello and welcome to May CRC. I am pleasured to introduce Matthew Indest from Curaleaf. He is going to talk a little bit today about what's going on with him, talk about his background, talk about what's going on at Curaleaf, and just share some of his vast general knowledge with us. So I look forward to having this discussion. After his presentation, and Deering, I may interrupt some, but I'll let you take it away, Matt. Share your screen and let's get learning.
All right. Good morning, Allison. Yes, I am Matthew Indest. I have a background in horticultural science and in agronomy, so focused on plant breeding and genetics. During that time, studied a lot from production horticulture, but also a lot of research focused. So while I was at LSU getting my degrees and such, that's where I was exposed to kind of the whole research process. Worked at different research stations in the state. Actually lived on site with one of them for about a year and a half, supporting various research projects. So always very interested in the science of plant cultivation, both in a horticultural setting and an agronomic setting. So I've had kind of the pleasure to work in both areas and really now have the opportunity to bring that into the cannabis space. So I'm excited to be working with Curaleaf and can share a little bit about that work. We can also dive into some of my other side projects, which you know a little bit about, Allison. Yeah, I guess it's been a few years. We met in at your field day. I think was my first time to meet you in person. And that was actually some of the first exposure to commercial scale cannabis that I got to be a part of. And so thank you again for that. And here we are now. So let's see, I can make sure I'm doing the right window here. Let's see, are we live?
We are.
Looks good.
Yep.
All right, so this is a slide deck, Cannabis Cultivation as an Applied Science. My title with Curaleaf is Technical Director of Agronomy and Plant Improvement. So that's quite a mouthful, but means I get to work in a lot of different cultivation processes and do this at a national level for Curaleaf. So for context, Curaleaf, if you don't know, operates in 19 states, 9 EU countries, and we have 150 dispensaries, and those numbers are growing constantly. So with this footprint, we support an entire house of brands that range from different SKUs of flower, concentrates, vapes, edibles, beverages, all the fun, new, exciting cannabis products that are coming out into the market. So you'll see some of these in different states, and yeah, it's great to see that portfolio expanding and our footprint as well. We are.
What is your favorite product?
Oh, my favorite product. So I'm really a fan of the hash rosins. They've launched that in the Florida market. So a lot of the new genetics that we've worked on are now hitting into those. But I've actually been really impressed with some of the distillate products and how well they've been able to capture sort of the flavor profile and the aroma profile that you get in a flowering plant. There's a lot of times you go into a room, you smell it fresh, and you get into a jar, you smell it dry, and then you get like a distillate cart or something that's named after it, and it doesn't quite hit, right? There's definitely a disconnect between what that presented as in a natural state, and then but I've seen that improved a lot, and so that's been really impressive. So yeah, I think for kind of the convenience and stuff, I'm a fan of sort of the distillates and vapes. But yeah, good question.
Awesome. Thank you.
Yeah, so to do all this, and like I said, all these different states, we've got people all over the place, and they're working hard every day for the goal of bringing good or great quality cannabis to our consumers in every market. So I have the honor to be able to work with all these great people who are day-to-day boots on the ground, getting all these things out into the market. So it's been fun, and just wanted to give them all a shout out for their hard work every day. So as you and I both know, there's a lot of value in genetics. That's kind of the most important piece, in my opinion, is as the foundation to a successful business. There's an intellectual property value there that, you know, is meaningful for operations, and without good quality genetics and pairing that with best cultivation practices, you don't get the results that you want. And that's a hard thing to support a business if you can't produce a quality product and do that consistently. And genetics are kind of the foundation for that. If we look at domesticated and wild type populations in cannabis, there's significant morphological and chemical diversity. And so what I'm looking for is things that are fit for purpose. And depending on the project, that changes. And in the breeder space, in the genetic space, not everything is created equal. So, you know, there's a lot of different things that you have to go through to validate whether these genetics are something you want to bring into your operation at all. And then even once you get them into your hands and into your facility, how do you treat them from there? So that's an important piece and something that I put as a high priority in my work as I go around and work with different people in the fields, is to understand what they're growing, understand the challenges, understand what their purpose for cultivating is, and try to marry all those together and make sure that those are well matched. So in the end, you know, we're really talking about cultivating trichomes, which is a fun area of research. There's been a couple different conversations kicked up on LinkedIn that, you know, are centered around trichomes, and some recent papers that have come out dissecting those, I guess, is maybe a good way to put it. But, you know, we're screening genetics at Curaleaf for those that are commercially viable into specific market categories. High cannabinoid flower, largely focused on THC, and very aromatic flowers. I want something that's going to grab the attention, be loud, as we say in the market. But then also, you know, there's other categories and cultivation methods that focus more on, you know, either solvent-based or solventless types extractions, like we mentioned with the hash rosin. And so the trichome morphology, the plant morphology, these all factor into the success of these different, you know, product categories. And yeah, that's, you know, it's more than just the plant, right? So we're looking that close, and we're trying to understand how these trichomes are developing and what influences that. So from my background in a different trichome paradigm was in the world of cotton. And for those that don't know, cotton fibers are trichomes on the epidermis of a seed coat. And during this whole development process over 40 plus days, the initiation happens on the seed coat, and it expands about an inch and a quarter over this life cycle. It's a single cell that gets that long. So what I'd studied back at my time at LSU and the USDA was different, was how that cellulose in fiber develops over this period of time. And what, you know, we knew is that because it's in the field, it's experiencing variation, all of those different environmental factors play a huge part in the end quality. So things like the length of the fiber, how fine they are, that all kind of compounds over the season into what you get as a final product. So when I came into cannabis, I'm looking at trichomes, you know, cannabis trichomes the same way. As they initiate an early flower and they begin to develop and really bulk up until that final maturation phase, where it goes from this milky amber to potentially even capsizing. You know, that was kind of front of mind for me, is what environmental variables and what things are we doing that are going to manipulate how these trichomes develop to make sure that we're maximizing their production and their concentration on the plant surface. So trichomes are cool. And so we want to look at like what we describe as the phenotype. This is the physical expression of the genetics in a plant. And this can be broken down into a lot of different ways. So I look at it as qualitative traits. These are things that have discrete categories, such as disease resistance or color, the cannabinoid profile. So like maybe the ratios of the cannabinoids to each other, or an aromatic profile. Does it have limonene or does it not have limonene? Those are things that are qualitative. It's sort of like yes, no, or high, medium, low sort of bucketing of traits. And you can see in the pictures to the right, that's just an example. They look similar in their bud architecture and the morphology of the plant, but they have a very different color. So it's just one, way that you can see that like they check certain boxes together, but they differ in their color. Quantitative traits are ones that occur over a distribution or can be measured over a distribution. And these are height, diameter, stem caliper, yield, the actual concentrations of the cannabinoids or aromatic compounds. And as you see on the right, you can see that distribution that is shown, in this example. So population A, maybe the distribution of cannabinoid profile is tighter compared to others. And so those are different things that we look at both on a population level, meaning a lot of different individuals, but also as we look into the individual is how, consistent is it in these traits. So when you look at what is comprising the phenotype, it's one, the genetics, but then how it interacts in the environment. So in a field environment, there's a lot more sources of variability that we'll get into. I'll list out in just a second. But as you go from one environment to the other, it could be favorable at a general level, or it could just be favorable to that specific genotype. So there's things like crossover effects where, yes, you may have a better environment, but if it's not suited to that specific cultivar, you could have what's called a crossover effect. So that genotype by environment interaction is something that if you're a field breeder, you want to stabilize. You want a variety that's going to either perform consistently across all environments, or will have a sort of an additive effect. So as the environment gets better, the performance of the cultivar gets better. So you have to kind of understand all these different factors, and in the end, what it relates to in terms of the physical expression of the plant. This is a picture from our fields in Colorado, and these were massive plants. So I just love to show off how great those looked that year. So to dig into the genetics, I mean, you could look at the different letters of DNA, the RNA that's transcribed, how that puts into proteins, and then what are those proteins modified. But in the end, we're looking at a plant. So to really understand at each plant level what that looks like, the use of molecular markers and genotype by sequencing sort of tools allow us to start to unpack what genes are associated with what traits, and then be able to leverage those in a breeding program, or even just a kind of a validation process. Like, is this variety the same as this variety? They've got two different names, but seeing two different results. So maybe there was a mix up in the clone, or things just weren't what we thought they were. So this is something that's kind of rapidly evolving for the industry, and something that's been interesting to start to apply in a commercial setting. So when we look at those outdoor sources of variability, these again are fields from Colorado, and it shows some of the nutrient analysis that was performed pre-season to help us understand what we needed to do to amend the fields. But in a situation like this, where you've got rows that go down the field and an irrigation system, and you can only manage so much, you're kind of limited in an outdoor environment to what's present. And so depending on the amount of variability from your high to your low, these things could have major impacts, and you'll see differences within the field as well as across fields if I go to look at different fields. And so all of this sort of compounds into what you see in a population as variability. So if you have a clone across this entire field, you could see different phenotypes because of these factors. And then if you have a whole population, if you started from, say, seed, where you're looking at two different individuals, well, now you've got to factor in, well, is this genetically different from its sibling, or is it some other factor? So not without plenty of variables to unpack. And that's the fun part of doing all this at such a scale, as we can kind of get a very high level view, see the whole forest, and really try to dig in on what's going on with each of our trees, so to speak. So outdoor sources of variability are plenty, and I've just listed a bunch out here. I don't think I have to go through each of all of them, but it all compounds into that multi-factor equation of genotype by environment, and leads to the end result that you get. So what I can say, again, this is just a little bit more of how all these things factor in. But at the end of the day, we're focused on, cultivating the best flower, largely smokable grade flowers that can be then fed into some quality extraction platforms. So in an indoor environment, I have the fortune of being able to control or at least minimize many of these different environmental factors. In an indoor environment, we're able to fix the amount of light. The media is generally kind of a blank starting point, and then we're adding feed, with each irrigation cycle in order to maintain the nutrient levels that the plant needs. So we're not limited by the variability in the field. Like it's hard to move the needle for certain nutrients in the field in a short amount of time. But in an indoor environment or greenhouse environment where we're mixing the media and we're supplementing in a prescriptive way, that allows us to have a lot more control and a lot more consistency. The same with all of our environmental parameters. Assuming that engineering is not limited, we're able to maintain temperatures and humidities in a very tight band in order to meet what's optimum for that phase of growth. So with that, we also couple it with cultivation practices, so horticultural practices, in order to manipulate the plant canopy in order to maximize flower yield, as opposed and get it all in a consistent zone within the room, so that the environmental parameters are as uniform as possible. We do this through steps of training, topping, skirting, defoliation, in order to make sure that there's enough airflow underneath the canopy and make sure that our flowering kind of zone is as uniform and as and is within a narrow band to maximize on both light and that temperature and humidity zone. So all that kind of comes together as a big concerted effort to grow the best flower. And with all of that sort of standardized or harmonized, then we have a platform which makes breeding a lot more effective. I can actually develop cultivars specific to the needs of that environment, and instead of trying to chase down some of the environmental stability aspects that you would have to do in the field. So this is why a lot of cannabis cultivators rely on clones, I think, is because we're able to control the environment. So a clone can be put into I can hand it off to a buddy and they can grow it in their environment. And as long as the environments are consistent, if they're keeping it in a normal temperature range, they should see a very similar expression. Now, if they go and plant it in one field here in Louisiana or one up in in Oregon or something, then the photoperiod response, all those factors I laid out, you may see some very different results. And I know you've done a lot of work on that, some of the G/E stuff in the hemp fields. That was impressive when I first learned about all that.
Yeah, still so much to learn.
Yeah.
So if you could pick, would you be only outdoor or only indoor?
So I would put that on fit for purpose. So I do have about 10 acres of outdoor production here in Louisiana, but that is all industrial hemp for fiber and grain purposes. I wouldn't try to do that inside with any kind of, scale needed. But for cannabis flower and, and for THC flower production, I favor indoor controlled environments for the reproducibility. And then in my experience, there's a lot of control or quality control issues or, concerns that happen in an outdoor environment. One, we talk about heavy metal accumulation in plants, but just even like wind blown soil particles can be enough to cause failures on heavy metals, depending on what the environment is. So I think in order to deliver the quality that's expected for the programs, medical and recreational, I think indoor or at least controlled environments like a greenhouse are going to be needed in order to maintain those quality expectations. So
do you think so right now when you guys grow outdoor, is that for smokable flower or is that just for extraction?
Great question. So historically, there have been attempts. We've tried to get flower from those fields or from the hoop houses, and it really hasn't been the most success. There's more issues with seeding. You're more likely to have microbial failures and things that just prevent it from going to market. And the density of the flower seems to not quite get there. And with some of the recent exits from states like Oregon, Colorado, California, the majority of our production environments are now indoor. There were some hoop houses in Miami, and those, we've stockpiled plenty of biomass for extraction. But the majority of it, we're largely focused on just producing the highest quality flower right now. Biomass is easy to find in most markets, oversupplied, easy to get at a low price, but high quality flower is what's in demand, and that's the primary focus at Curaleaf. So
this is just general question. I know you guys are in so many states, so I imagine you'll have however many states you're in, you'll have that many answers. But say you did produce an outdoor crop and the flower had heavy metals. In theory, with distillation, it should leave those heavy metals behind, right?
So I would leave that for a chemist to give the best answer. But I can say that in my experience, there are contaminants, like pesticide drift, that do carry over through certain extraction platforms.
Right.
And there may even be residual amounts that aren't detected or aren't fails for your flower, but through certain extraction platforms, you do end up concentrating those, and they persist and then show up in finished product. So that's a risk that's not really something we'd want to take, and do a lot of testing to make sure that doesn't happen.
Sure. Yeah, just a random thought. And I guess my question there was, do they test the flower or do they test the extract? Or both? Both.
Yeah, right. And that's where I was saying, so you may have some that's like a low level, and then through an extraction platform, or you buy a third party and things look clean, and then in the oil it's hitting for something. So what specific platforms are better able to remediate that, or what carries over in which one? I'll defer to a chemist.
Okay, I'll mark that down for Marcus Rogan. We'll have him on soon.
Oh, great. Like I said, my doctorate was in agronomy, worked with cotton, but largely focused in breeding and actually getting into horticulture. That was kind of what brought me in. I met a professor at LSU who had a tissue culture lab, and he was the fruit and nut breeding professor. So he invited me in to work in his lab. We started working with stevia, and then I learned about all the other cool things he was breeding, here in the state, and kind of jumped in. That's what sucked me into the horticultural realm from my previous pre-med route. So with that, I started breeding in sweet potato and did that for a year and a half, learned, that whole pipeline, which I think there's a lot of parallels to draw there. It's another vegetatively propagated crop. So that's a parallel that I look at a lot. But breeding's always been of an interest to me. Once, you have like a very good cultivation practice, like you need new, fresh genetics. We see it in all of our crops. It's kind of a constant, like what's new, what's latest and greatest. And the cultivation practices, I think, kind of stay a little bit more fixed after you've dialed those in. Like, I don't want to have to like reinvent my feed recipes for every cultivar. So I try to find ones that are best fit for the middle of the road, and then find new cultivars that can improve in their optimization and utilization of those resources. So that's what breeding represents, is kind of this continuous improvement over populations and over time. And it's something I spent a lot of time at university studying. So when I came into cannabis, new crop, hadn't been, explored in the same way as literally like splitting cotton fibers, and getting down to that level. It seemed like there was a lot of opportunity for genetic gain, and really just finding the cultivars that could be plugged in. I mean, even just sifting through what was available. So that was exciting for me, and being able to do this with Curaleaf, we were able to start, a small breeding project, just proof of concept, feminized pollen, taking our best cultivars in the Florida market and rehitting it with the other best performers, and then start to sift those out. I like this scheme, and I am very interested to know if, as people talk about feminized breeding, what their experiences are. I've heard mixed tales of caution about whether feminized breeding is right or not, and whether you're compounding problems or you need to be hunting males. But for me, I see it as an opportunity to select on the traits that I'm interested in, and make quicker genetic gains. But I do acknowledge the risk that there might be an opportunity for populations that are prone to intersex through stress, that could be carried on if you're not attentive in selecting away from that characteristic. If you have a plant that looks to the left, is this a herm, or is this a induced, I know because I applied the STS and we had stress test these clones prior to performing this process. But without that, if you have a plant that just will produce male pollen under stress, then that could be carrying over, and then you're inadvertently selecting for those characteristics. So segregating populations, once we've done these crosses and start to look into them, we're going to grow them in large number. As somebody who's taken a lot of statistics, I appreciate large population sizes and understanding as I'm trying to recombine two traits from distant lineages that I want to check as many boxes as possible in my selections. So I need to see as many individuals as possible to improve the chance that recombination event will happen. So, we're focused on trying to fix those target traits, and any of those genetically controlled traits are going to be limited to the diversity that's in that germplasm pool. If I'm looking for PM resistance, I'm not going to find it in plants that aren't PM resistant. It's probably very unlikely that those new alleles or traits are going to just pop up by random mutation. So we want to find what's in the population now, and then start to bring that into some of our elite lines, and just continue to just check more boxes off as those go, and make that sound easier than it is. But it's part of the fun process. So with any of these breeding projects, we start with what idiotypes, what is the ideal type? What are those series of boxes that we want to check? And then define what parental combinations are going to get us there the quickest. And then, everything cascading after that. How big of the population, how many populations can I look at? But it all starts with defining our end goal. And this could be things like a shorter flower plant height for indoor, something that maybe doesn't require trellis. We're always looking to increase the amount of flower yielded from our plant, like jarrable flower, not just total biomass. And then potency and unique organoleptic profile. And then secondary to that, we start to look at how it performs in different extraction platforms. So one of my better performers down in Florida was a cross of GMO and Purple Punch, which was named Modified Muffins. Fortunately was a great hash yielder, high potency, and it was one of the parental combinations in a lot of these crosses for that reason. We're striving for more potency. We want things to look frosty. We want things to hash well. And this is just one example of something where we found what we were looking for, and then now we're trying to introgress those characteristics into some other varieties that just they smell great, they look great, they have something unique, but they just don't have the yield and the potency that we really want. So we're trying to bring those traits together, and we'll see that come to fruition with some new drops. And then one of the other advantages to breeding, besides just getting us traits that we're looking for, is related to diseases and pathogens. Plenty of people are talking today and are around the industry about hop latent viroid. It's probably the most impactful disease that I've experienced in indoor cultivation. And so every clonal propagation after that first set of cuttings is an increased risk of pathogens and stress accumulating in the plant. A lot of people talk about genetic drift, but as a population geneticist, that has different meaning to me. What I see in these clonal propagations is not like a change in the genetics, but a change in the expression. So the genetics are fixed. We have clones that are moving around, but it's that genotype by environment sort of interaction. Their environment is like this diseased state, right? If they've got this hop latent viroid, it's impacting the way the genetics are expressed. So what I've seen in the field, these were two different cultivars. A healthy individual was testing at 20%. Another healthy individual was around 18%, but with symptomatic for hop latent viroid, we tested, and those plants were testing in single digits to like 11%. So very high loss of cannabinoid yield. That's THCA yield in that specific example, and that's obviously very impactful to a cannabis operation if you can't produce the trichomes that have the characteristics you look for. These are some pictures that show some moms. These were healthier moms. It's possible they were infected just given the proximity, but these are severely infected to the point where you're seeing the symptoms in your mom stock, and that's not a good thing. So get these out, get them removed, test all of your remaining moms, test frequently, test routinely, and make sure that you're starting with clean plants. Looking at the literature, at least 2 years ago, there was the idea that seeds would be a lower risk, if not possibly even a way to get out of dirty plants. But through testing of crosses from different breeders, including myself, we've seen transmission through seed. And that can be detected as early as the cotyledons, or the true leaves, and those rates are there. So it indicates the need to have healthy plants to breed with before doing this, because you will see carryover. And I've seen rates as high as 70% in 2 different seed populations with hop latent viroid at this phase. So seeds are not perfect, and there's a lot to be done to try to clean up hop latent viroid. So
is that even with seed coat sterilization, or is that without?
Yes. So that is with it. So that's showing that the tissues inside the seed are potentially infected. And I think there's been other people who have kind of given that anecdote as well. But yeah, with seed surface sterilization with like bleach and stuff, you can, it'll look clean on the surface, but then once you look at that, maternal tissue in the seed, I think that's where it's carrying from. So I'm not sure if it was pollen borne and that's what brought it over, but I think more likely is that the maternal tissue that's part of the seed is carrying that over. And based on what we've seen in tissue culture cleanup, like it makes sense to me, is you have to get down to very, very small, undifferentiated sort of cells before you get to a cleanup point.
Right.
And these just
I've heard a lot of back and forth on this, on the medias.
I think we will. Yeah, I think we'll continue to hear.
Oh, for sure.
Yeah.
And I've heard people say, it's just something we're always going to have to deal with. Just keep culling. And I've heard people that, clean out their whole facility a few times because they're trying to start fresh.
Right.
What do you think?
Yeah. Well, so yeah, I've seen and experienced both. I think there's a need to kind of rip the Band-Aid off and get clean stock in as quickly as possible. I think it's something that we need to be continuously mindful of and continuously work toward, because you can get to a low presence. I have not gotten to the point of eradicating. It's not like any, there are facilities that are clean, but looking at a national scope, we're in the single digits. But again, like if you become relaxed and somebody stops following some of the sanitation SOPs or anything like that, you can easily transmit from one mom to the next to the next. And we've seen evidence of persistence in runoff, not specifically infection, but at least persistence that might suggest that it's possible. So restricting cross transfer between plants and runoff is one practice. That's one way that we mitigate the risk. But it's not gone, right? And I think I'm only 2.5 years into this whole process of understanding, hop latent viroid and cannabis, and we've made some great strides. I don't see half of a room stunted the same way that we used to, or the level of symptoms that I showed in that cannabinoid profile. But it does. Sorry, kids are home for the summer.
No problem.
We're definitely moving in the right direction, and I think a lot of it's going to come down to technique, using tissue culture appropriately, looking at the sensitivity of your assays is really something I've spent a lot of time on. Is you've got a lot of different options for testing, PCR methods, qPCR methods, some LAMP assays, some different like immunostrip type things. And they all have a varying level of sensitivity. So if you're not able to see it when it is there, then it could persist for a lot longer, and you get a sense of comfort thinking things are clean, but those could be false negatives, and that's not the type of error that you want in an operation, because then you have symptoms and you're like, well, we're clean until we're not. So
Yes.
and these are just some fun pictures from the whole breeding pipeline. So I'd just love to show those off. So
Side note, I've been doing a lot of work with feminized breeding, and it's frustrating between varieties. I have some, say I'll have 5 different varieties, and I'm spraying the same concentration on all. And some will just, it's throwing as much pollen as a true male would. And then there's some where, you're lucky to have one male pollen sac, basically. Have you seen the same?
Yes. Yeah, there's been a lot of different things that influence the pollen, production rate. And I think there's certainly the interaction, between your silver product and, the concentration, the frequency that you apply it, when you start to apply it. Those are all things that you have to hammer out and understand what works. First phase of, these pods was taking all those good parents and spraying all of them. And, it was like 30 or so different lines or, clones that were all sprayed just to find which ones would produce pollen. And some of them would produce the flowers. So you would get this kind of mixed, phenotype here. And then sometimes they wouldn't open. So the humidity in the room can affect that. The humidity, can affect the pollen shed as well. So there's a lot of things that go into this, and it's different than just like, growing cannabis. But for floral production, we're now trying to grow for seed. And so there's a lot of things you kind of have to tweak here and there, and to make it successful. But yeah, there's definitely a variety influence on the effectiveness of those sprays. But I've been happy actually. I've had some autoflower crosses that those were successful, and I haven't been in a case where I wasn't able to get pollen out of the ones I needed. If I really wanted it in the cross, then it was on the receiver side then, if it was harder to cross with. So
Interesting.
But yeah, I gotta give a shout out to the team at University of Connecticut and their publications on some of this stuff. That was very helpful in getting started. A lot of the university research that's out there has been very impactful in my work. As you come into cannabis and you're like, as I was complaining about not having enough to do, like enough exciting, interesting things in this world of, conventional agriculture where everything's already, we've redone all these fertility trials every year for 50 years. I come into cannabis of like, oh, there's all this stuff that could be done. And then you get there and you're like, but there's no playbook at all. Right. So you're writing the script. So it's, you got to take the good with the bad. There's a lot of opportunity, but there's also not a lot of references to build off of. So
For sure.
Very appreciative of all the other, universities, Clemson being one of them, that have been putting priority focus into helping cultivators of cannabis and trying to build to that body of knowledge.
Yeah, and I appreciate this group and folks like you and Travis coming on, and
Yeah.
you know, of course, everybody has a little bit of intellectual property they gotta keep to the side. I mean, that's commercialism, but sharing a lot of the very basic things that you think you would find in a journal. Us being able to talk and share is, I think it's really important and awesome that you can do that.
Yeah, no, that's why I love, I love interacting with people like yourself and, other scientists that are out and about at different conferences and kind of just in a very casual way talking about what we're working on. And like, it's been very validating. As much as, there are some trade secrets and things like that, the community's been very willing to share. I try to share as much as possible. I think it's beneficial for everybody. And what I found more often than not is like, there's a lot of validation there that like, we're all kind of working on the same things. We're doing things pretty similarly. And there is some nuance there that you really have to do that at every operation though. That's what I've experienced. It's like, we have like standard harmonized practices and things defined, but when you go to the different operations, there are some considerations depending on how they're set up, where they are in the process of getting to those standards. And, it's kind of meeting people where they're at and then building from there. And it just takes time. So the more we've shared, the better it's just been, in getting things going. So.
For sure. So when you came in 2 years ago, did you find it it'll be there one day. Did you find it difficult or easy to start implementing some of the practices that your background and training saw that some of your facilities needed?
So I would say there were some challenges. There certainly were. There's sort of like legacy sort of ideology, and not all of that is bad. It's not. I would say a lot of what I was up against is understanding like the justification. So I would hear we're doing X because of Y, or in order to do this, and that logic didn't always stack up. What I would see though is the practice itself wasn't necessarily bad. It was just more the logic and the justification behind it that didn't always make sense. But with all of these sites, that was the first, I guess it's still kind of going on, is what are the little practices and things that we're doing at all these different sites that make them unique, and identifying what those best practices are. So one of the first tasks was understanding our fertilization program nationally. What does that look like, and what is the right way? Yeah. And then migrating over toward a common feed recipe, or an adaptive feed recipe that could be tweaked to specific needs. But first, like, what is that foundation? So going in, Curaleaf growth by acquisition model means we have cultivators in different sites across the country that we're operating, and then we brought them in. And so there was a lot of different feed, different brands of fertilizers, different ratios, different supplements, all being kind of added. And it was up to the fertigation managers at each site to kind of develop those recipes and, do that. So coming from a very technical background, how do I make sense of this? I've got a part A, a part B, I've got this one that's like 3 different things, and then they're also adding this. So for me, it was, let me get a spreadsheet together and went site by site. Here's week 1, 2, 3, 4, 5 through 8, 9, 10, and what are you feeding them this week? And then literally documented across like 15 of our sites to say, here's the PPMs that they're delivering every week, across these feed programs. And it was very interesting. I have all that mapped out. And so seeing how some sites would tweak, nitrogen in different ways, or potassium and phosphorus, and you got to have more CalMag, or, whatever it is. It was just being able to map all that out and then, figure out like, where's the common ground here?
Mhm.
A lot of leaf nutrient analysis was done across that time as well, and seeing like, is what I'm seeing in the field a nutrient deficiency or some other symptom?
Right.
Then compiling kind of combining all that nutrient analysis to build our own reference ranges for what those targets should be. And there was, again, some great papers out there that start that work. And it's, I think a lot of it's focused on vegetative phase of growth. There's some newer ones coming out in the flowering side. And so I've used both of those literature references as well as our internal references to kind of be guardrails for anytime we do leaf nutrient analysis. And with that, we've developed a kind of a national feed program, and we're in most places have adopted that completely, but there's still a few that are, either using up their fertilizer reserves that they've purchased and, making sure we're using that inventory. But so far, I mean, that whole blend has worked really well. And yeah, proud to have been a part of that whole development process and to see it actually being successful,
Right.
kind of your big.
I mean, across all those platforms too, I imagine you were able to make a really solid conclusion for upper management to see, whatever metric you want to use, price per square foot per week of fertilizer costs. I imagine there were some really big differences there that,
Yes.
Just a little bit of science and spreadsheets can say, look at this difference.
Right. Yeah. And so that was a big factor too, is cost per gallon or cost per gram sort of thing. And yeah, some of the fertilizers, I mean, coming from, the background where like salts are cheap, like it didn't make sense to me why this fertilizer was this much. And at the same time, a lot of people were wanting to experiment. I mean, it's but you have to have that background, I guess, to do it in a way that's cost effective and that can be leveraged at the national level. And what I'd seen is a lot of sites would buy something, or they would get samples of something, they'd try it, and then they would see a little bit of improvement, and then they would just start adding that to the program, but they wouldn't necessarily measure it in a controlled way to make sure that ROI was reproducible. Or they would order a bunch of something and then stop using it, and then you've got a stockpile of product X on the shelf.
Right.
So I think getting to this like foundation of here's all your basic nutrients, Goldilocks zone, not too much, not too little. You shouldn't see any problems. Call me if you do. We'll troubleshoot that. And that's gone very well. So now there's this next level of, are there any other supplements that we could be using? But we had to start from that foundation, and make sure, that we were at least covering all of the bases before we start to try to, throw more things at it.
Right. How interesting. That's just so many groups to work with. So
yeah, that's the fun part too, is, everybody's got great ideas. And it's helping them work through, okay, if that's the question we want to answer, how do we develop a process that gives us the answer, like that can tell us, is that true or not? And so that whole hypothesis, question, testing, I bring back the scientific method as often as possible, and just like, hey, let's just start here. Like, what are we asking? And are we asking the right questions to answer, to get the answer that we're, needing here? Because if we're asking the wrong questions, then we're never going to get to resolve the problem we're trying to fix. And, it's been very validating with everything that we've learned in school to then be able to apply that in such a large scale fashion, and to have it be successful. So I enjoy it. Certainly enjoy the job.
All right, I got two more questions for you. One,
Cool.
What are your hobbies?
What are my hobbies? Well, if you look behind us, we got some more plants.
Of course.
The tomatoes in the backyard have been coming in pretty strong. So my interest was always in medicine, and and I wanted to be an anesthesiologist. got interested in like drugs, I guess, as a student of DARE in 5th grade. And they came to school and they're like, hey, like all these things like cause your body to have these different effects. And it was just like very interesting to me that like you could take something to change this. But I've always been interested in plants and plant derived medicines. I thought it was really cool that like you could grow something that, you know, had a medical benefit or recreational benefit. So I've always been interested in studying things like that. Like, there's a fun one that I think you might know about, is buzz buttons, and they make your mouth tingly, and you know, it's very, very fun sort of novelty sort of, I guess, how would you describe it? Pairing with like a drink. And so I've been growing a lot of those. That's been fun, and a lot of hot sauce. So all still plant focused. You know, we grow so much weed with Curaleaf that I don't need to home grow.
Yeah, you've had enough.
Yeah, I've had enough. That's all work. So here we grow a ton of house plants, a lot of succulents, a lot of, you know, yeah, just things that have like a unique, novel characteristic in the plant world.
That's
Got any
awesome.
fun, cool plants that you could send my way? I would love to.
I do. I've actually got some seeds I'm gonna send you this week. So
Great.
yeah,
Great.
I will. It's also most horticulturalists I talk to, their hobby is also plants. I mean, mine certainly is. I have the same sort of, you know, kind of excitement around plants that can manipulate the body in some sort of way, like the buzz buttons or miracle berries. Have you heard of miracle berries?
I need to try those. Yeah.
Oh, that's so cool. Yeah. Right. So for those of you who don't know, it looks like a little coffee bean when you harvest it from the plant. Really slow growing shrub, and it's only hardy basically down towards Florida latitude.
To grow it here then.
Yes. So it would do really well for you. I have to bring mine in the winter, but anyways, these little berries, you let them, they taste nice. It's just sweet. You let it kind of sit in your mouth for a little while. And what happens is it makes your taste buds perceive anything that was sour to be sweet. So you could literally take a lemon and stick it in your mouth and it would taste like lemonade. I mean, it is crazy. Or a tomato that's super acidic, it'll taste like sweet salsa or, you know, something like that. But it's really wild how and why. Super intriguing. Yeah. But that only lasts 30 minutes. Thank goodness, because it definitely would ruin meals. But for a party or just having fun, it's that sort of thing.
Yeah, I love pulling out a little bag, and people always first thought is like, because you know they know what I do, they're like, oh, he's got a little bag of weed, and it's like, no, it's a buzz. And just like oh yeah, that board weed, whatever.
Right. I
was like, that's all, like that was last year, right? Like this is the newest thing. So yeah, plants, and I think, you know, because of plants, like I also really like to cook. That's kind of a big, you know, I try to cook as much as possible. And largely around like, well, I have all this stuff that I'm growing, like the intention is to eat it. So combining all that stuff into new recipes and stuff, it's been a fun thing for me. So, and as much as I like to eat, it always makes sense to actually make something you like, right? So that's all. I think that covers my hobbies for the most part.
Nice.
The kids too.
Okay, and then one question. What has been your favorite or most interesting experiment, lesson, whatever you want to call it, that the CRC has put out?
Oh, that the CRC has put out. Wow. Wow. Well, now you put me on the spot. I was going to, there have been so many good ones. You know, I make sure to pay attention every single time they drop. I think the photo period one is very interesting. And there's a lot that can be unpacked there. And then I think over the weekend, I saw a lot of the, you know, attention over the fertility management for height restriction. So in the way that I operate, as I digest literature, I see these ideas out there. I look for opportunities in our operations to then validate that and see how we can apply that at a larger scale. So there have been numerous opportunities to do that. And again, I would say it's very validating to see that all continue to work, in the field and really understand how to apply it. I think what we've unpacked in a lot of ways is like there's a huge cultivar dependent aspect to your rooting hormone ones, your your photoperiod stuff. And so then the challenge becomes like, yes, we see this response, and which cultivars can we apply this to?
Right.
Where does it make operational sense? So that's where things really get dicey, is how do I plug this into an operation and have all the scheduling that goes and how that changes things. So
Right.
yeah, so we have to balance the need for consistency and the opportunities that these different adjustments can make. And so it's really exciting to see how that comes through. And as the national menu sort of develops, having that documented to know like this is a photoperiod response cultivar that has this, or this one favors this type of rooting hormone, like getting all that documented and shared nationally is definitely high on my list. So I can't say any specific one that was a favorite, but there's certainly a lot of gems in there. And
well, it's really nice to know that you're taking some of these ideas and taking them a bit further commercially, because that's pretty much our goal, is to take some of these crazy wild ideas, which aren't really crazy wild ideas. I mean, if you look at photoperiod, Dr. Faust himself has done a bazillion DLI studies and photoperiod studies on poinsettia. And so It's just plugging in this as the host plant. But I get it from an operational standpoint. You've seen some of the pictures of photoperiod studies where if it's at 16 hours, the flower looks terrible. I mean, it's just spindly and something that is a lot of money for a Curaleaf to do on a 1,000 square foot area. Whereas let us kill our plants and make really bad flower, and hopefully give commercial cultivators, a parameter to work around to start optimizing their facilities too.
Right. Yes, that research cost is high. And again, if you don't have the right people doing it and documenting it, then, it's a lot of, potentially wasted effort, or misguided, and when you get to the results. But working with people like yourself and, that's been a huge value. And then, kind of consolidating that. So like I said, with the fertilization part, was like everybody was kind of being their own little scientist, but they weren't isolating all these other variables, right? You've got other people in the grow also doing that. So being able to look at that singular variable, and then now, kind of running with what you guys have shown, there's certainly been some opportunities that are being capitalized on, and certainly, looking forward to everything comes down this year. I know you've got a lot of stuff in the pipeline.
We do a lot of more exciting stuff to come out, and I can't wait. Let's talk a year from today again.
Sure.
See what you're doing. See what you might have used. See what's going on. I'd love to catch up annually if you're down for it.
I'm always happy to talk.
All right. Well, thank you so much. We will talk to you soon.
All right, Allison. Thank you. Have a good day.